All right, so now I'm going to take you through a few of the applications that I mentioned here. Spectral fingerprinting first. And so spectral fingerprinting takes advantage of, again, the white light laser and the tunable emissions that you see here with the ability to move the laser lines left and right and move these gates left and right. All right, so if we can collect every excitation for a sample and we can collect every emission, what if we do both? What if we collect every combination of excitation and emission? Well, first of all, that gives us a lot of data. So we can collect, in a typical scan, we would collect 1,500 images. That'd be every combination here where emission is longer than excitation for the reasons that we talked about before. And we can take that information and plot it, and that will create a what we call a lambda squared plot, or I consider it to be a spectral fingerprint for for the image, as well as every individual pixel in the image has its own spectral fingerprint. All right, so let's see what this looks like. So here's a sample of an egg chamber. For speed purposes, you can do these at somewhat of a low resolution, then it might just take about a half an hour to run the scan. If you do high resolution, it could potentially take you several hours to do. So this is a bit of a lower resolution image, but the purpose is not to get a great image here. The purpose is to get 1,500 images pretty fast and then be able to plot their intensities. So here what we've done is we've drawn a region of interest, which is this green line. It may be difficult to see, around this sample, this egg chamber from Drosophila. And what we see here in the spectral plot, this is excitations over here and emissions in nanometers. And what we see are there are two dyes in this sample, and that's good because I put in two dyes here. I also put in DAPI, but DAPI is not part of this because it's over in the UV. So this is from 470 to 670 nanometers. All right, so there are two dyes and they're well separated. And by knowing their spectra in both directions here, you can decide exactly where it is best to excite and best to collect this dye. Now we also have spatial information here. And in fact, as I mentioned, every pixel has 1,500 values. And so every pixel has its own plot. What we can do is draw a region of interest around some feature. For example, here, the cell borders here, the cortex of the cells where there's lots of actin. That's our green stain. And so we just draw a box or a region of interest around a few pixels here. We can get then just the fingerprint for that region. And you can see our actin stain with its green dye is confined mostly to that region there. There's just a little hint of the other dye. If we switch and draw a region of interest around a couple of nuclei, then we see our other dye, wheat germ agglutinin, with its far red dye, and that is most abundant around the nuclear envelopes here that we've encircled, and then that has very little of the actin. So this lets you get spectral information on any region within your sample. So that can be very useful. This is especially useful if you have lots of autofluorescence, which we don't in this particular sample, but often there is a lot of autofluorescence and you can sort it out using these types of plots. This is just the software setting that gave us this image here, and that was 1,500 images. We're stepping through every 4 nanometers in excitation, every 5 nanometers in emission, and that we can change those. We can do a very low resolution scan a lot faster if we want to do it that way as well. I'd like to show you a quick application now of the lambda squared plots, and this, for me, this really highlighted the power of this approach. So we were working here with Taylor Sewell from John Sedbrook's lab. They were looking at plant seeds, and they had a wild type and a mutant plant seed. And so we decided to try and see what the difference is between the wild type and the mutant. We did this lambda squared plot. Now these seeds have not been stained. They just have lots of different autofluorescent compounds that they store up. And so in this particular region here, we drew an ROI. And so in this particular part of the seed, there are a large number of different compounds, and two major compounds or sets, related sets of compounds here. But when you look at the mutant and choose the same region anatomically, what you find is that those two peaks are almost completely missing, and yet the other compounds are still present. So that's a way of really, in a very detailed way, biochemically characterizing your mutants to see exactly what is different. All right, so for our next imaging mode, we're going to talk about FLIM. That's Fluorescence Lifetime Imaging. I'm just going to give you a quick overview, give you a sense of what's going on there. So why do we want FLIM? So FLIM adds a third dimension to our images. So our traditional imaging has two dimensions, excitation and emission. But we might have some compounds that have very similar excitation and emission characteristics, and they're probably not going to be identical, but they might overlap to a significant extent. Then you'd have a hard time separating those two dyes. But if we add a third dimension, that would be how fast the photons come off of the dye, independent of their excitation and emission wavelengths. There now we'll have a three-dimensional plot, and that gives a much better chance of separating those two signals. Even if they overlap on these two axes, they probably don't overlap very much in terms of lifetime, because that's an independent parameter for that molecule. So just as a reminder here, there's a timing difference between when the excitation light hits the dye and when the emission light leaves. And it's very fast, but it is measurable. And so our detectors can measure this timing and then calculate the timings coming from each emission event. And that gives you a decay curve. This is a bit like radioactive decay. You have a certain half-life that before the dye gives up its photon as an emission. And based on this, the computer will calculate a value called tau, which would be the typical lifetime, typical timing before the dye gives off its photon. So we can look at that here in terms of what the system is actually doing to collect this data. So this only works if you have a pulsed laser, because you have to have a defined start time. And so our white light laser is pulsed, so it will give off pulses at some constant rate. You can also use the 440 nanometer that's also pulsed. So after a pulse hits the sample, then the detector will count how long it takes before it gets an emitted photon following that pulse. So let's say there's 5 nanoseconds, and then the hybrid detector detects a photon that's come off of the dye. And after another pulse, we collect another photon, and then it will count, let's say, 2 nanoseconds, another pulse, let's say 8 nanoseconds later before the first photon hits. Now you could swamp out the system by having lots of photons coming in, so you want to dial it back so you only get maybe 1 or 2 photons per pulse. So all of these timings are collected and plotted, and that's what gives you this type of a decay curve. So most of the photons will come out early, but then this will tail off. Some of the photons will be a bit slower to arrive. So then we can calculate tau, and then we can color code the image spatially based on the lifetimes. So blue will be dyes where the photon comes out very fast, and red will be dyes where the photon comes out more slowly. All right, so here's what that does for us. If we collect our spectral information very widely, so we're just collecting all the emissions that come out, we're basically going to collect a monochrome image. So we're just taking all the spectral data, basically throwing it away, just saying, okay, how many photons do we have per pixel, regardless of their wavelength. And so that's this side of this image. So this is from the plant seeds that we talked about earlier. Now if we switch on the FLIM module, then we start to collect timing information, and that provides a lot of contrast. There's a lot of timing differences across these different fluorescent compounds in this seed. Again, there are no stains here at all. All of this is inherent chemical differences in the different compounds that are present there. All right, so that is what it looks like in plants, where you have lots of autofluorescence, but almost every organism will have some type of autofluorescence. And so here's a fly example, a Drosophila example. This is the thorax of a fruit fly right here. And we have GFP expressing in some of the muscles. And so here is the green channel that we're collecting. There's nuclei over here and transmitted light over here. But this is our green channel. And the problem here is we have our GFP, but we also have the cuticle. And the cuticle of the fly, the surface of the fly, is also fluorescing in the green channel. But it's a very different compound. So if we then switch on FLIM, this is what we see. So our GFP shows up here, and it's green. This is just coincidentally because that's how we're color coding our lifetimes. So there's our GFP, but now our cuticle is a very different lifetime. It's much faster, and so it shows up as a different color. And you can actually, if you look closely, you can even resolve the GFP and the cuticle, even when they're right up against each other, basically spatially not separable. So this is a very powerful method for sorting out different compounds. This can also be used to detect changes in a compound, because the fluorescence lifetime depends on various features of the environment. It could depend on the pH, various other things. So if you're observing in a live situation, observing differences in lifetime, that means that reflects something that's happening to your dye. And in particular, this is a very good way to perform FRET, fluorescence resonance energy transfer, that is used to determine how close to, for example, fluorescent proteins are to each other. As they come closer, they're able to transfer energy without using a photon. And that will greatly change the fluorescence lifetime properties. So as two things come together, the lifetimes will change, and then you can observe proximity of two proteins.